before a brief break will be given by Dr. Lihong Wang, also from Tsinghua University. The title of her talk is The Neural Correlates of Trait Rumination.
Oh, change the title. Yes. Actually, I just changed my title. Hello, everyone. It's such a good pleasure to be here. I would like to thank Mr. and Mrs. McGovern, and I really appreciate for their great contribution to this neuroscience field, and I'd like to thank McGovern Institute for inviting us. What I would like to talk today is about rumination, stress, and depression vulnerability. Should I?
We didn't connect this. Choose the
Oh, okay.
up and down buttons there.
Okay, thank you. So, as everyone else in this room, I'm really, really curious about our brain. All I would like to fascinate is about whether we have intrinsically in our brain have a network wired, and that network has been changed by our experience, and how that is changed ours between you and differentiate between you and me, and how that change when we get to the mental disorders. So my study actually mainly focused on the neural circuits related to emotion and cognition. But today I would like to talk a little about depression vulnerability. Actually, all the words I put here are my interest, my interesting words, keywords that I'm interested. I think everybody here knows about depression and knows how diverse it is to us, to our life. And one thing I would like to mention is in China, there's only 1/3 of patients go to the clinic. And there's a high ratio of suicide every year, but many of them probably have depression. So it's really important to detect who are vulnerable to depression as early as possible. But for the vulnerability of depression, I think this is a very nice review paper by Nortif, by Dr. Nortif's group, and it has risk genes based, and also changes in neurotransmitters, and also the brain structures. And the important thing is he here actually grouped 3 types of phenotypes. If you have the increased self focus, probably you have the changes in the resting state activity. Or if you have maladaptive emotion activity, and that might be increase your stress sensitivity. Or if you have your reward induced activity reduced, then you may have anhedonia. It's interesting that when I would listen to Dr. Lu's talk, I think maybe someday we can have this photo tube to stimulate our dorsal raphe. I'm not sure to treat. Anyway, today I would like to focus on 2 phenotypes. One is increased self-focus, the other is increased stress sensitivity. Actually, I would like to talk about this to emphasize 2 things in this neuroimaging field. I think as a neuroimager, we are in the very exciting and challenging era, because there's so many new methodologies has been developed in doing data analysis, and particularly for the network brain, and all the old concepts is changing, and it's called paradigm shift. Like the Brain Initiative, we want to have a large data set to really to find out what's the stable signal from our brain. But I would like to say, while you're looking at this large data set, what's important is to looking at the commons between them. Today, I would like to talk. One example is this: you're looking at different states of the brain, and looking at how in that area whether there's some common findings. So then you can using that kind of information to do the support machine learning, so that you get a good feature for them. That is what I'm talking. One of those is increase the self-focus rumination. Self-focus, which is rumination in depression. Rumination, actually, when we're talking rumination, actually, we can't forget this scientist, Dr. Susan Hoxema. Actually, she is the pioneer, and she has built up lots of work in looking at rumination. Rumination is a mode of responding to distress that involves repetitively and passively focusing on symptoms of distress, and possibly and on the possible cause and consequence of the symptoms. So it's really focusing on the negative side of self. And rumination does not help for problem solving. Instead, just focusing, fixated on the problems without doing any action. So that's people who have rumination predicts the depression onset, and also who have the rumination can stay longer on the treatment. I mean, not responding to treatment that quick, and also have recurrence of depression. Dr. Nolan Hoxema, actually, she developed the ruminative response style. This is a questionnaire. We use that to looking at how you're having higher or lower ruminative function. So there's, in the literature, I think there's a studies like looking at, well, when you inducing people to in the ruminative status, what the brain activation look like. So the task is we asking you to think about what people notice about your personality. On the contrary, the contrast is think about a roll of shampoo bottles of display. Since this bottle of display, this bottle is more concrete image, so they develop another contrast, which is abstractive sentence. So think about what contributes to team spirit. So they're looking at rumination versus control condition, and they found that in depression, they didn't have those decreased activation in the subgenuine cingulate or rectus, actually. On the other hand, rumination versus abstract sentence is actually showed greater activation in this hippocampus, amygdala, I'm sorry, and parohippocampus and PCC area. So these are the conditions when people were induced this rumination activity. But typically, because this is in a block design, and you may not always focusing on this condition, so there are some limitations of the task. On the other hand, Dr. Rieto from Carmen Arshner's lab, they're looking at, well, your reappraisal, your emotion feeling. So the task is to give you a cue of increase or decrease, or just looking at those pictures. And then after the cue, you show the negative pictures or neutral pictures, and asking you, so then you have to reduce the emotion. And by self-focusing, like if you enhance the emotion, you self-focusing on this picture, or you reduce the emotion by are we thinking of this emotion as somebody else and nothing important? And then you're asking how you have your affect change. By looking at this, they found that the rumination actually is correlated with the anterior cingulate in the condition of just looking at the pictures versus decrease the emotion. And so this ventral medial prefrontal cortex, I would say, is related to the emotion regulation. So then these are in the normal healthy subjects, so then they were actively coping with this emotion. So we would like looking at just resting status, like we say, during whether you have, if you are sensitive to depression, maybe you have been wired and have the highlight intensive of the connectivity during the resting state. And I use this, there are several methods of looking at resting states, but one of this is using the seed-based connectivity. So what seed-based connectivity is you just looking at one region to looking at up and down of the waves, and then looking at what other areas of the brain have the similar synchronized with this area. So that's called functionally connected. So we used several of those regions that has been proposed related to emotion, self-focus, and memory and executive function related to depression. And to looking at this depression association with rumination trait, rumination with the functional connectivity, we're looking at the young healthy adults under tryptphen depletion. So which tryptophan is the precursor of serotonin, and after you give the 15, typically you have 16 balanced amino acids, which is important for our amino acids. But when you reduce one of the tryptophan, and then by competition, you have low tryptophan, and through the brain, you have low serotonin. So this is pretty quick. It's after 3 to 5 hours, you see the peak. You have low serotonin in your brain. And so we're looking at just like Dr. Luz has just said, that serotonin is related to depression. And so this is one way to looking at serotonin effect. And we wondered that when you have a temporarily low serotonin, what's your brain, whether those who have higher rumination traits have changed or not in their functional connectivity. And the other is looking at the older people. The reason I'm looking at older people actually is thinking, if you have this rumination trait, but you don't have depression, so that might be you have to have some protective in your brain. But on the other hand, who have developed versus those who developed depression. So this is the experiment about this acute tryptophan depletion. First, we have the control condition. We give people 16 amino acids, and after 5 hours, 3 hours, we do fMRI study. And versus we give them tryptophan, 15 types of amino acids by liking of tryptophan, and doing the fMRI scan. So actually here we have those emotional tasks and also decision rewarding tasks. We didn't find any significance regarding the decision and the rewarding, actually. But today I was just only looking at talking about the resting state functional connectivity. So this just showed the, after 3 hours, the tryptophan level becomes lower. And we did fMRI during this low period of tryptophan low period. So this is we use the seed based. So this just show one seed and PCC area, and the yellow colors are the activations of the connectivities from the PCC area with the PCC area, and we just comparing the lower figures, which comes from Fox et al., and it's just saying this are really mimic the study. And we found that in those depressed patients, not depressed, I'm sorry, those who have higher rumination actually showed greater change from connectivity from the PCC to middle frontal gyrus area. So this is the connectivity change between the tryptophan depletion versus a control condition. So this, after the tryptophan depletion, they have increased connectivity between PCC and middle frontal gyrus. As you know, the PCC is the region of default mode network, and middle frontal gyrus is test positive network. Typically, these two networks are negatively correlated, and this is just saying under acute tryptophan, you have enhanced the opposite way of connectivity between the two network. On the other hand, we also looking at the association with anxiety. Actually, it shows differently. So the anxiety actually associated with the increase the connectivity between PCC with amygdala area. So I'm not sure this will be generalized or not, but this is just differentiating at some point, differentiating depression versus anxiety. So we're also looking at rumination in older adults, and this, as I show in the next figure, actually those in those older adults that don't really have much of the high rumination score. But it's picture looks like this. It's just as you have greater connectivity, after you have the higher rumination score, actually you have lower connectivity between PCC and MFG area. This is just the opposite to that, to tryptphen depletion. And on the other hand, in depression, it doesn't have any of those connectivity, just doesn't associate with rumination. And so this actually removed the effect of depression. On the other hand, we found that from the seed of amygdala, it showed the greater connectivity with the subgenuine cingulate and the MFG and fusiform gyrus area in patients. So this just shows the difference between you have temporally reduced serotonin versus you have depression. So in healthy subjects, you have negative connectivity between PCC and MFG area, but when you temporarily reduce serotonin level, you have increased connectivity between you have increased connectivity between PCC and middle frontal gyrus, and also in depression you have increased connectivity between MFG area with amygdala. So this just showing an example of when you're looking at those different mental status, that will give you some feature of whether there's common area related to rumination or not. So another talk I would like to address here is this increased stress sensitivity. So as we know that the support vector machine learning is helping, everybody right now is looking at using this method to differentiate, to giving us a classification diagnosis, hoping that someday to help us to do the individualized diagnosis. But one thing I would like to mention is so far is across the publications, the classifier they found is only stays with that data set. It doesn't come across with different data sets. So that's why I mentioned previously. So if we use the features that we demonstrated from different data sets or different mental status, then that will help us for the individualized diagnosis, give us a better feature for classifier. On the other hand, we probably can using the dynamic change pattern to looking at for the individualized diagnosis. So this actually, I would like to talk this using this example of increased stress sensitivity. So as you know that the depression often comes with you have a acute stress or worse event and you have depression. But actually it's not everybody have depression after acute stress. Actually you have this genetic risk or early life stress, and then you have this ruminative nature and also the negative bias versus somebody who are resilient for depression and maybe have positive bias or mindfulness. And my hypothesis is that if you have repeatedly receive a stressor, for those who are resilient to depression, to stress, maybe they show the the activation like this in their emotional regions. So they get increased activation, but gradually habituated to it. So I called it habituated emotional response. On the other hand, for those who are vulnerable to depression, they may gradually increase their activation in their emotional region and stay plateau or sensitized to it. So that's what we called prolonged emotional response. So we're looking at whether we give subjects this repeatedly giving that the stress, whether we can find this pattern in the brain. So one thing I'd like to mention is this early life stress, actually, which is a vulnerable factor for depression. So Dr. Taylor actually did lots of work in documenting this early life stress and later life mental disorders. And she also found that in those who have family who have been raised by risky families, and then how they had actually reduced activation in the amygdala area, which is quite opposite to what we thought, but actually it's just because of the long-term the stress they have really inhibit the activation in the amygdala. So what we used is looking at this give subjects repeated the stress task. Subjects just doing mental mass stressful task. This has been validated by many other labs that is reduced stress. We're looking at this repeatedly doing stress versus repeatedly doing mindful task. Because we're looking at the change across time, we our interest in looking at the first run response versus the last run. If the first run has greater activation than the last run, we think this is a habituation effect, or maybe it's because it's the cognitive change. On the other hand, if subjects show the pattern like this, I would say this probably is depression vulnerable. I have to say this has to be in the emotional region, not in the cognitive control region. So this is the self, on the left side is the self-rating stress, and it's obviously during the stress mental mass task you have shown have greater stress rating than the mindful task. And also subjects showed relatively increased heartbeat than the mindfulness task, but not breathing. And interestingly, this is younger normal subjects. So then, in general, overall, we found that in many areas in emotion related to emotion, like in amygdala and hippocampus or fusiform gyrus, there all showed habituated response. And we didn't find the opposite side at the group-wise, but interestingly, when we're looking at the correlation with the early life stress scores, it shows that those who have early life stress, higher early life scores, actually showed increased activity in the not increase, it's decreased activity from the first to the last run, and particularly that's associated with the emotional abuse. So I have mentioned that we use this mindfulness tab, but I didn't mention this, the measure that we use. Actually, we're also looking at the trait mindfulness, which using this 5-facet mindful questionnaire. It's about observe and describe, act with awareness, non-judgment of inner experience, and non-reactivity of inner experience. So these are defined how you're mindful or not. And if this mindfulness actually, one of them, we're looking at the correlation between of those 5 facets with the rumination, with rumination, and we found that only this non-reactivity showed correlation, this negative correlation with rumination trait. And we also looking at, we differentiate our subjects by looking at who are vulnerable to depression by if subjects have early life stress or not, and if they have, and whether they have a rumination tendency, and also have low mindfulness. So if so, we define them as depression vulnerable. On the other hand, if subjects have early life stress but have high mindfulness and low rumination, we think probably they have depression resilience. So we're looking at the pattern of those two groups, and we found that in the depression vulnerable subjects, in the amygdala area, they actually showed pattern like what we predicted. So they showed gradually increased activation in the amygdala, but stay plateau. And on the other hand, in the ventral medial prefrontal cortex, which is related to emotional regulation and negative correlated with amygdala, actually shows from the first two, the later two runs, they show decreased activation. On the other hand, for those we define as depression resilient, show the opposite side in the amygdala, they show gradually decreased activation across runs. But on the other hand, in the ventral medial prefrontal cortex, it's just keep having the high activation across all runs. And for those who doesn't have early life stress, and so they showed not typical of what we can characterize. But interestingly, if you can see that for the depression in the amygdala area, in the vulnerable depressed, depression vulnerable subjects, actually they don't have a greater activation than those who are resilient to depression in general overall. But it's just a fit for this pattern. So by showing this, I would like to say that if you're looking at the pattern across time, that might give you something about individualized diagnosis. We're also looking at how the negative non-reactivity protected with us from stress, and we found that this is a no-go task, and in this, the red and the green are the activation to negative go and negative no-go. As you can see, they're overlapping in some area in the insular and inferior frontal cortex area, and those showing that this area actually responds to negative per se. And we found that in the, those who have non-reactivity of mindfulness actually showed opposite negative activation in the insular area. So, which suggesting that there doesn't respond much to negative response, which probably is a protective feature worth to further explore. So to summarize, I would like to say that if we're looking at common findings across different mental status, that will give us a good feature of a disease. And if you're looking at dynamic changes pattern in fMRI signals, which can allow us to have individualized diagnosis. Those are actually all the work, as I mentioned, actually was done at Duke, and I just moved to China. And we are continues doing the same study, but we want looking at who are with the genetic and also this rumination characters. Thank you. So this is the picture of Tsingua in the spring. So welcome.
So I'd like to ask you what you consider early life stress and whether or not an individual resolves that stress could make a big difference in terms of how it affects them earlier. So some people who have a very stressful young life and overcome it can't be swayed at all, right? And then I'm not sure what you mean by mindfulness. So do you take account of, first of all, different kinds of stresses, and what are the determinants of mindfulness?
Okay, so actually this mindfulness, so I'm just starting from the back. The mindfulness is what we just based on that five facet. The measure actually is when you're observing. So what we have, the last we think is important to us, not having protect us from depression is this: when some stressful events coming, you don't respond immediately. It's non-reactivity. That's how it's defined. But so right now there's lots of mindful-based treatment, so therapy. That's teaching people to actually focusing on your. I think the ideal situation is your mind is out of your body. It's mind not associated with, right? Don't aware of yourself. So that is the condition of training. And this has lots of studies showing that you can, after the therapy, you can increase your attention and cognition even. For the early life stress, actually, it's increasing. Like there's also several features like physical abuse or emotional abuse and emotion neglect, and many of those. It's also we have those just a questionnaire to fill out. And there bunch of literature that show that it can last to affect your later life. But as I showed here, some of those who have early life and can get aware of it and have this mindfulness, I think that might relate to some other later life experience and also self, cognitively adaptation, I think. So that's is very important.
Maybe I can follow what Marcel just asked about mindfulness and rumination. So on your table, you define people with early life stress either as vulnerable versus resilient. And in that, you use rumination and mindfulness as two things, observed measures. Do they always go hand in hand? In other words, if you have scores low in mindfulness, does it always show illumination, and vice versa? Because someone could be, having illumination, and yet having a having a higher mindfulness, right? I mean, score in terms of it doesn't have to be going hand in hand. What if you have a situation where someone has rumination and yet high mindfulness?
Well, that's a good question. I think what our data shows is show the negative correlation. But I don't think I can absolutely exclude make the two. It's quite opposite. But theoretically, yes. So you can if you have good observing and not responding, and probably you should have lower ruminative feature. But that may not 100% not happening though. It's worth to looking at in larger sample. So we just have a very little sample, very small sample. Yes.
I think actually my question may be related somewhat to yours. I noticed the two large personality factors you had up there were negative affectivity and positive affectivity. If you look at the Tellegen multidimensional personality questionnaire, which has been linked to genetic studies of twins, that also includes conscientiousness. And then each one of those factors is related to subfactors. And I find myself thinking, both as a clinician and as a researcher, that rumination, in fact, would be good for some people, but it might be not so good for other people. So it would co-vary as a function of personality traits. So that's the first thing, and we know there are biological underpinnings to that. The other thing is that there are studies out there that show that depression, in fact, improves reality testing for some folks. So I wonder if what we're talking about is a relationship between stressed out people who have negative experiences that are encoded in the amygdala in a particular epistemology. I mean, that's cognitive behavior therapy, which is only one way of thinking about therapy and thinking about personality. So I guess I'm wondering, would it make sense to look at personality traits and how they co-vary, and also to begin to think about the advantages of depression? And by this, I don't mean a hypomanic approach to stuff.
Right. So you're right that rumination is just one aspect of it, and is also we can looking at negative bias and other like neuroticism. Actually, we did encode the big five personality, but I didn't do because I don't want to do multiple comparison. I just didn't do the analysis yet. I think it's worth looking at.
Yeah, I think the big five is a little different than the multidimensional personality.
That's right. That's right. Yeah.
For a variety of reasons, especially in terms of the biological research and the genetic research.
That's right. Yeah. It's worth looking at.
All right.
Thank you.